Alright, so hi guys, I'll be presenting a research paper by Wang et al. Published in AMB Express. This is entitled Ammonia Oxidizing Bacteria and Archaea within Biofilters of a Commercial Recirculating Marine Aquaculture System. And I am fulfilling this as part of my requirement in DF 304 or Advances in Fisheries Research. Using a metagenomics approach, the authors of this study looked into the microbial communities in biofilters of a commercial grouper aquaculture system. And they look specifically on the ammonia metabolizers. Again, to those who are not familiar with the term metagenomics, it's a broad field in molecular biology where you study genetic material recovered directly from environmental samples. So, you can expect in this study that DNA fragments recovered from samples are used to characterize what species of microbes dominate in the biofilters of your RAS, or again, your recirculating aquaculture system. Cloning and sequencing of the AMO-A gene showed that nitrifying bacteria included this species of Nitrosomonas. Okay? And of course, 15 strains not associated with the described species. So this is what I'm talking about. They utilize a target gene here. It's the AMO-A gene. And they use it in the identification of the nitrifying bacteria. So only those species in the sample capable of oxidizing nitrogenous compounds are expected to have this gene. And aside from bacteria, again, not only bacterial species are found in the samples. And not only bacterial species possess this gene, there are some archaeobacteria that were also detected. And take note that archaea are different from your bacteria. They belong to a different domain. And for your nitrifying archaea, these are the included strains. 8 strains of Nitrosopomilus. Some undescribed congeners and other undescribed archaea. So it's dominated by your Nitrosopomilus. So that's it for the abstract. Let's have a few background first, or background statements and concepts before going into the methodology. Now, effective biological filtration by ammonia oxidizers is critical to fish production in recirculating aquaculture system, or RAS. And an enzyme unique to your chemolithotropic ammonium oxidizing microbes is ammonia monooxygenase, or AMO-A for short. And this enzyme oxidizes ammonia to your intermediate hydroxylamine. Again, ammonia is toxic to fish, and we should allow this bacteria or microbes to break down ammonia. And this gene or this enzyme that is encoded in the gene becomes our sort of ID to only select the ammonia oxidizers from our samples. So those microbes that were included in the samples that do not have this gene are not included in the analysis. And for grouper production, it was usually carried out in flow-through systems, in highly intensive culture ponds, and more recently in recirculating aquaculture systems. So take note that recirculating systems are theoretically the ones who will make good use of biofilters, because we need to filter the water before returning them back to the system. And previous studies on this area showed relatively frequent observations of Nitrosomonas sp and Nitrospira. So in most biofilters that were examined so far, these are the usual genus that shows up. So it will soon be revealed if in this study, or this study will be consistent or is still consistent with this trend. Now let's go to the materials and methods. Let's take a look at the RAS setup here, where the study was conducted. So we have here 16 tanks, each having 7 meter in diameter. So you can imagine how big these tanks are. Each, of course, is an inlet and outlet for recirculated water. And you can check the numbers here representing the critical components of the system. So you have initial filtration here in the board screen, okay, to remove your large solids here, okay, your board screen. And then water gets collected in this sump here, number 3. And it's pumped into the foam fractionators to remove the fine solids. Okay. So you have number 5, your foam fractionators. Then you have a series of biofilters here. Okay, 6. Here. These biofilters are made up of elastic polyolefin media that treats ammonia and other metabolic waste products. So you leave the filtration or the removal of ammonia to the microbes that are found in the biofilms in these biofilters. Okay. Then after passing these filters, you have the UV. Okay. You want to get rid of vegetative forms or your heterotrophic and coliform bacteria. And finally, you inject pure oxygen here, and before returning the water to the system. Okay. Let's proceed. We have for the fish culture setup, we have 9,200 hybrid groupers. So 1,200 per tank. They were stacked into the RAS for 240 days from on-growing to harvest size. And the picture I'm showing here, again, are not the actual tanks. This is only for visualization purposes. But as I have mentioned a while ago, they have 7 meter diameter, large enough to hold 1,200 fish. Okay. And here are the initial data. You can pause this vid and take a longer look at this. Okay. And from here, 30 to 50 fish per tank are randomly sampled monthly to be able to determine the body weight gain, length gain, your specific growth rate, and feed conversion rate as we are culturing them in the span of 40 days, 240 days. And total ammonia was analyzed weekly using the finite method. And for the nitrite analysis using the NED method. So basically, these are more into titrimetric analysis of water samples. Okay. Now, for the characterization of microbial communities, DNA must be extracted. Okay. So you extract DNA here. And they should be amplified via PCR, okay, and sequenced, okay. So here we have our biofilters cut into small pieces, and the microbial film was suspended in your phosphate PBS or phosphate buffered saline solution. This is to isolate your DNA from the cellular debris to lyse your cells. And you have you centrifuge and you remove the supernatant to collect your DNA. Okay, and you do some quality control of your DNA extracts by allowing it to pass through a spectrophotometer. Then your DNA samples were amplified, cloned, and sequenced using your PCR machine. And these are the amplification conditions here for your PCR and the primers that were used. So it's the 491 base pair fragments for your bacterial AMO-A genes and the 635 base pair fragments for your archaeal genes that were amplified by a PCR. And we continue in this slide. We have the purification of your replicate samples. We purify the target genes or the target sequences or target fragments using your gel electrophoresis, and they cloned it later to get more samples. So about 10 positive clones from each sample were selected at random for DNA sequencing. So you have your DNA sequencer here. So the purpose is to for you to be able to create your phylogenetic tree. And you can use bioinformatics or your gen banks to look for established genus or species. Okay, so you have to create this phylogenetic cluster analysis to recognize microbial community diversity in your samples. Okay, so again, after your PCR, you sequence and you identify what's in your sample. Let's proceed. We now go to water quality, fish growth, and production. Okay. So this table shows various mean values for water quality, fish growth, and production. Again, you can pause to have a thorough look. These results here show that the series of biofilters metabolized ammonia effectively as the nitrite and total ammonia nitrogen stayed at these low concentrations here. And for the growth performance of your hybrid grouper, the researchers also met, the RAS met the expectations for commercial production, because from low starting density, if you can still recall, it's 6 kilograms per cubic meter, we reach these final densities of 52.3 kilogram per cubic meter. So that's a lot. It's amazingly an increased final fish density. Okay, more than I expect at this point. So from 6 to 52. And researchers also reported that no disease outbreaks occurred and no antibiotics or chemotherapeutic agents were being used in the production period. And the survival was almost 94%. So that's really good. So the RAS is very efficient. And let us now look at the bacterial populations detected in our samples. So we have 31 unique bacterial AMO-A gene sequences that were observed, and mostly within the genus Nitrosomonas. And again, not all DNA are recognizable. The researchers observed 11 distinct sequences from different strains of Nitrosomonas that could not be associated with a described species. So when you encounter things like this, unidentified, because you lack information in the database, these perhaps are open to new names if they are identified as novel. And there are also 4 unidentified groups that could not be related to unknown species. But because of their high similarity with other Nitrosomonas sequences, they seem likely to represent lineages within that genus, within the Nitrosomonas. Okay, now we go to your archaeal communities. Phylogenetic analysis showed that the archaeans clustered into 7 taxonomic units, and that most of the sequences were homologous to those of Nitrosopomelo species. Now you can see here the various strains observed, and results also include 6 sequences from an unidentified archaean, OTUs, or groups. So as noted above, the inability to associate environmental DNA sequences with known taxa is again not surprising, because you will always expect that in metagenomic studies. The database is not yet complete. There are still lots of unidentified groups that are yet to be uncovered, or shall we say named and described. And DNA sequences for this group, for the confamilial Nitrosarcium sp, this was grouped within the lower branch of your Nitrosupomilo species. And this was somewhat unexpected. But again, this can be due to perhaps to too little sequence having been analyzed. Maybe the samples are too small to be able to resolve through phylogenetic relationships, or perhaps in the database there's still unresolved taxonomy in the archive database. Again, as I've said earlier, the database are not yet perfect at this time. So that's it for the results. Let us now try to consolidate everything and discuss the important aspects or findings in this study. Now, for this study, numerous Nitrosomonas species and strains dominated the bacterial community. So again, we have Nitrosomonas for your bacterial community that dominates, and for the Archaea community, it's dominated by your Nitrosupomilo species and strains. And these results contrast somewhat with those of earlier studies, at least in part because of the methodological advances. And in the paper itself, if you read the paper, the authors mentioned various studies from 1996 up to 2017. So from 1996, where metagenomics still relied or is on its early stage of development, it relied only on limited databases, and up to the present. So here I can no longer present these findings here. But again, the main point of reviewing these studies by the authors is that they want to point out that because of a more updated database and the use of this AMO-A gene here, this study was able to identify more species than past studies. So meaning they have achieved a greater ability to detect and characterize ammonia oxidizing microbes, because the researchers build upon earlier work by making use of better developed DNA sequence archives. And perhaps they were also able to associate more clones with particular species, especially those whose environmental requirements are now better known. So, all in all, this study uncovered a diversity of both described and undescribed species. And I will present some of them here. Okay. Now, 3 different strains of Nitrosomonas europaea was observed. So this is a microbe, first isolated from soil and common in freshwater environments and sewage. It doesn't require salt. And also another sequence was observed from N. stercoris. This species occur in strongly eutrophic environments such as wastewater treatment plants, and this indicates, of course, that they contribute to removing high concentrations of ammonia, because again, in highly eutrophic environments, you have lots of metabolic waste there, organic wastes. It's highly concentrated there. We expect ammonia to be in high concentrations in these areas. Also, we have your cryotolerance, which is also found in the samples. It's a cold, tolerant, obligate halophilic bacterium. So it's a salt-loving bacterium. that was first isolated from Alaskan marine waters. And there is one observation of N. eutropha. It's a bacterium that is found in highly eutrophic environments such as sewage disposal systems. And like your N. stercoris, as I have mentioned a while ago, it also tolerates elevated ammonia concentrations. And N. eutropha can grow anaerobically. Okay? And finally, we have two N. estuary strains, which is common in marine and estuarine waters. And also 8 different strains of N. marina, which occurs in marine waters and salt lakes. So, for the observation of a diverse assemblage of Nitrosomonas and other ammonia oxidizing bacteria with differing optimal ammonia and salinity concentrations, this suggests that ammonia oxidation can go forward under a range of environmental conditions. So, microbes can perform ammonia oxidation in different environmental conditions. So it doesn't select a specific condition. And this allows for the diversity of microbes that can oxidize ammonia or that can metabolize ammonia. And this diversity also suggests that ammonia will be oxidized effectively in different microhabitats within biofilms throughout the treatment or treatment train or treatment runs. So again, because you can perform ammonia oxidation in different conditions and environments, so this increases the efficiency of your biofilters. Also, the presence of ammonia oxidizing archaea. Okay, again, not the bacteria communities, the archaeans. It also suggests adaptive differentiation because archaeans can have their own niche, and their activity can support niche partitioning in the natural environment. And for the archaea communities in our samples, there are 84 archaeal clones included. Your Candidatus maritimos, Candidatus synarchium. And one study by Sakame et al. Identified only N. maritimos among the 63 distinct clones. So a previous study was only able to identify one, but this study showed 2 strains of that species, and including 3 named and 5 unnamed congeners. So those that are similar to that species. And another 6 unknown archaeal OTUs. So meaning this study was able to describe more than one compared to a previous study. And analyzing the biofilter nitrifier community in a marine RAS producing shrimp, in a marine RAS that produces shrimp, another study by Brown et al., they found that archaeal genes were more abundant in all compartments of the RAS than your bacterial genes. Okay? But in a different study, Bagchi found low archaea diversity. Okay? So, I think these are considerable differences among studies. So, even those using similar methods, these differences may be attributable to the environmental conditions, system operations, or source of seeding. Okay? So, sorry for the noise there. It's raining outside. Okay, I'll continue. And again, the differences in the findings can also be due to your environmental factors, such as pH, oxygen concentration, and temperature. And all of this will affect the compositions of your microbial communities that is responsible for nitrification. And both AOB and AOA are sensitive to a number of environmental conditions. So the dynamics of nitrifying organisms in response to nutrient removal and Water quality, I think it still remains poorly understood. So it is somewhat necessary in the future for us researchers to explore and better understand how the bacteria and archaea respond to environmental disturbances that can affect the water quality and a diversity of aquaculture systems. So it's again in RAS, it is safe to hypothesize that our biofilters can work as long as there is no disturbance. It's not yet known if they can still work efficiently if the system is disturbed. But of course, we control those conditions in an artificial system. And there is one author who proposed that your archaeans or AOA might be important factors within the nitrogen cycle, especially in low nutrient, low pH, and sulfide-containing environments. So this suggests the possibility of, again, niche partitioning within your aquaculture biofilters. So maybe in some parts of the biofilters, you have more archaeans there than in some parts. And this can increase the resiliency of the biofilter, because if there is a disturbance and you can disturb one species, there are still other species left to continue the function of the biofilter. In one study, bacteria or AOB can metabolize ammonia better than AOA or archaeans. But archaeans are 1,000 to 3,000 times of greater abundance than your AOB in marine environments. So this can suggest that archaea may be the more important nitrifiers, especially if you consider the numbers. If organic particles are abundant in your RAS, their presence may also affect the rate of nitrification and the microbial subcommunity that can thrive in it. Alright guys, that's it. I hope you followed through my presentation and gained a better understanding of RAS biofilters, grouper culture, and the use of bacteria or microbial biofilms in your biofilters. And again, you can read the entire paper for more detailed discussions by the authors themselves. And thank you very much for watching.